We will pay special focus on Cyclin D. First and foremost in this picture, I want you to come down to the bottom and where is this RB phosphorylation and focus upon the RB and the E2F complex. It is important that you memorize E2F and RB as being together. That complex, as long as it exists, will then properly regulate the cell as it moves from G1 to S phase. Your first arrest point that you will be focused upon will be between G1 and S. You know everything about that arrest point first and association with its functioning and the regulators, then you move on to second arrest point, which will be in between S and G2. Now, with this E2F-RB complex being present, then the cell is not permitted to move from G1 to S phase. Is that understood? How do you remove and how do you free up the E2F so that the cell can then move from G1 to S phase? The term is phosphorylation. Whenever RB gets phosphorylated, you remove the breakers. RB, you remove the break. How? By phosphorylating. In biochemistry, what is the name of that enzyme that is responsible for phosphorylation? Kinase. Good. Would you take a look at our topic, Cyclin D? CD word K2. What is the K stand for? Kinase. What does it do? Phosphorylate RB. By doing so, what have you done? Remove the break. Who is set free? E2F. Who is truly set free? The cell can now move from E1 to S. Therefore, what does this cancer want to do with RB? Does it want to phosphorylate it, or does it want to be dephosphorate it? It wants to phosphorate it so that you can remove the break, so that the cell remains within the cell cycle internally. What is the complex that you have memorized between Cyclin D and CDK? Which one? It's CDK4. You must memorize Cyclin D, CDK4. I want you to take a look at the complex in the picture. You will notice Cyclin D, Cyclin K, CDK4 is the active kinase responsible for phosphorylation of your RB, so that E2F allows the cell to go from G1 to S phase. You move through exactly to how I'm instructing you. You will be in fantastic shape. Once you have understood that, then you will take a look at the other cyclins. When was the last time you have heard of Cyclin D? I have mentioned this a few times. You have memorized Cyclin D as part of your mantle cell lymphoma T1114. At this point, it should make to you that if you are to upregulate Cyclin D, which will mean that you have increased the activity of CDK4, increase the phosphorylation of RB, increase the removal of the breaks or the tumor suppressor gene, and the cell was in your cell cycle. Isn't that cancer? In this case, obviously mantle cell lymphoma. In addition, take a look at the sign of the mutation above Cyclin D in the picture. We have mink and loss. Above that, we have growth factors, and the big one that you definitely want to know is PDGF. So ultimately, a Ferrari is a Ferrari. The back, the front, the side, it's all the same car, but how do you know it is a Ferrari? Because you are accustomed to the car and you know all the different angles. All I can do for you is give you the car and the various angles, but can I guarantee that I can give you every single angle? No, but just about. So here, what I'm saying is if you have excess growth factor, what kind of effect is that going to have on Cyclin D? Upregulated. Upregulate. If you want either C-mink, N-mink, L-mink, whatever it may be, those are different types of cancer. What do you want to do with Cyclin D activity? You want to increase it. It is all part of your signal pathway. At any point in time, if you have memorized this here, you are going to miss a question because you haven't understood what the dynamics are if your cell cycle. I hope that's understood. Let's continue. In this illustration, there is one more point that I want you to focus upon, and that is going to be your P53. P53 is also a tumor suppressor gene. Instead of dividing into details first listen to what I have to say as for its overall cell cycle. Whereas RB gene and its tumor suppressor activity will be specific with a rest point, one between G1 and S phase. Ultimately, that P53 is responsible for will be two major functions. If a cell has been stressed and if the DNA has been damaged, but the cell can still be repaired, P53 will induce your DNA repair gene. Is that clear? And if the DNA can be cleaned up or it can be repaired, then the cell will go back into the cell cycle and it will continue on its way. Function number 1 of P53, repair the cell. Function number 2 for P53 would be now if the cell has been damaged beyond repair. So now you have gone beyond the point of return. The cell cannot be, it doesn't deserve to be in the cell cycle. Why would you want to have a mutated cell in your cell cycle when you know that it could increase the risk of cancer? So therefore, P53 will do the right thing. It will take the cell out of the cell cycle and execute it. Welcome to apoptosis. So whenever you have a cell that has been beyond the point of repair, therefore, P53 is going to include apoptosis, and that will be through releasing cytochrome C from the mitochondria and therefore bring about caspase activity. Welcome to apoptosis. We will talk about that soon enough. Now, I told you earlier that I will take you one step further in anticipation as to what the boards will be asking. I want you to take a look at the very far end of this illustration with P53. And P53 first and foremost, yes, technically those work through P22. Not going to walk you through the detail, but let me ask you this though. If P53 wants to hold off a cell from going from G1 to S phase, listen closely to what I'm saying and what I will be asking. If it monitors your cell quality and P53 finds out that the cell is not worthy of moving forward, Now, what is that p53 going to do with Cyclin D CDK4? It wants to arrest a cell between G1 and S, does it not? So therefore, what kind of activity would it then exert upon your Cyclin D CDK4? Did you say inhibition? Of course you did. Very good. It will inhibit Cyclin D CDK4 so that you do not phosphorate your RB. You do not remove the breakoff of E2F. This complex will do what? Will prevent the cell from going from G1 to S phase, and in the meantime, p53 will be busy making sure that it is recruited the enzyme to bring about apoptosis. Is that understood? It is only after you have understood that point we can start looking at the specifics here. Now p53 could, and it does, detail-oriented with p21 inhibits your Cyclin D CDK4. Do you see that line there? That's a block. On the other side, there is something that I wish to mention here because I am getting feedback where this also is being asked. At least be able to identify INK. INK means inhibition of K kinase. What kinase are we dealing with here? Good. Cyclin D CDK4. I told you earlier whenever you find the letter P as being a prefix before a number or letter, it indicates P53, P16 is a tumor suppressor. It is normally going to control and regulate. It will inhibit the kinase. What if you had a cancer and inhibits P53, P60, 16? You inhibit the inhibitor who's activated Cyclin D CDK4, thus bringing about phosphorylation. Lots of important information that I have just delivered. Make sure that you have crystal clear with what I have given you. It is only then you move on to the next aspect of my explanation. How now there is something on the very end where we have MDM2 and you have your PG in there as well, but I don't want to go into much detail. Just know that P53 also through the help of MDM2 can then bring about feedback type of regulation. We are going to expand further and look into cell cycle so that you clearly see what I'm referring to. Let's begin at the very top. One. Whenever you see the word P in front of the number of or letter, the function of that particular element is to bring about tumor suppressor. And this is good things, aren't they? It's just when you lose complete tumor suppressor activity, it is when a patient is prone to different types of cancers. Apart from RB and P53, I will talk to you more. And another one is called VHL, von Hippel-Lindau, which is, of course, dealing with renal cell cancer.